Welcome back, students. In this section, I'll be talking to you about we'll go take the molecular techniques much more. So the next technique that I'm going to talk to you is southern blotting. So what is this southern blotting? From where did this? So does that mean there is a northern, eastern, western blotting? Actually, there are, but the first southern blotting technique was named after a scientist, Edward Southern. But later on, just as a spoof, the other names also have come. Now, southern blotting, what is it? It can detect mutations in DNA. It is a blotting technique for DNA. Southern blotting is for DNA. First, what is done is DNA are isolated from cells, maybe like WBCs. This DNA, which has been isolated from the cells, is cleaved using restriction enzymes. The cells, the different fragments that are obtained by the cleaving of restriction enzymes, they are separated by electrophoresis, using electrophoresis. So when they are separated, different size fragments of different sizes will migrate to different levels. The smaller fragments will migrate more. The bigger fragments will not migrate. So thereby fragments of different sizes can be separated out. So that is done by electrophoresis. Then in the same gel, the DNA is going to get denatured and it is transferred to a nitrocellulose membrane. This gel is going to be transferred into a nitrocellulose membrane. There is a particular method. During this method, from the gel, the DNA will go on to the nitrocellulose membrane. So this is called as blotting. Blotting, that is why the name blotting. It is transferred to a nitrocellulose membrane and then probe is used to identify the DNA fragment of interest. So maybe from WBCs or anything, we have the whole DNA. This DNA is going to be cleaved into smaller fragments using restriction endonucleases. Now, once it is cleaved into this, they are subject to electrophoresis. When you are subjecting them to electrophoresis, they will separate out. So they will separate out to different fragments. They will keep on moving, and the ones which are separated out by electrophoresis, we are transferring them onto a nitrocellulose membrane. You got from 2 people, we can get it done for 2 people also. These again, this is known as blotting, where the DNA fragments are this, and then a probe is used, and the target DNA is identified on the nitrocellulose membrane. Autoradiography is also a part of it, and then finally we get the bands that are present. So this is known as southern blotting. Now if mutation was there, as I said, this is used for DNA analysis. The restriction fragment, what can happen? Either it may result in a bigger fragment or it may result in a smaller fragment. So the target DNA may be either become bigger or smaller depending upon the type of mutation that is there and whether that restriction endonuclease is going to bypass that mutation or it is going to cleave at that mutation. So you may get a smaller or a bigger fragment. Now always this is also compared with that of a normal person so that we can come to know where the mutation is. So it is compared with normal and the mutation can be detected. So restriction enzyme may not cleave, it will result in larger fragment. If it cleaves at a new site, it will result in a smaller fragment. Now just as similar to this, there is one more technique known as western blotting. Now western blotting is used to detect proteins. So in this there is electrophoresis of protein followed by detection with an antibody against the protein of interest. Again we will get something similar to this. Blotting is used, nitrocellulose membrane is used, and we get the protein of interest. Northern blotting is also there. Southern blotting is for DNA, western blotting is for proteins, northern blotting is for mRNA. So for mRNA, so again in this electrophoresis of mRNA followed by hybridization with a specific probe to know how much of this mRNA is present. We do northern blotting. Now I go on to one more technique known as restriction fragment length polymorphism, or also called, abbreviated as RFLP analysis. Now what is this RFLP? Now genome varies between individuals. The genome content of my genome will be different from another person's genome. Each one of us individually will have different genome. Then how come that we all are not affected with the diseases? Even if a single mutation is there, we say there will be disease. So this variation that is happening is usually in the non-coding region. So this variation between the genome of different individuals which is happening in the non-coding region is called as polymorphism. Otherwise, if it is in a structural gene, you call it a mutation. Polymorphism is clinically harmless, does not affect the phenotype of the individual. So they are usually in the non-coding regions or the introns. So remember, so much of DNA, nearly 90% of the DNA is considered as non-coding or having introns. So when the DNA is subject to cleavage by restriction enzymes, the length of fragments vary because of the polymorphism. So the same DNA thing, when DNA of two individuals are there, suppose there are DNA of two individuals, that length that is got from one individual will be different from that of the another individual. Okay, why? Because there are this length and of another person may vary at a particular site actually, but let us look at like this. So one individual will have different length, the other individual will have a different length of DNA fragment when cleaved by the same restriction enzyme. Why is this? This is because of the polymorphisms that are present. So in this we have got two types, and that is, one is called as single nucleotide polymorphisms or SNPs. So in this, in the non-coding region, it is a single nucleotide that is different in different individuals. So it will lead to a change in the restriction enzyme cleaving site. That is SNP, single nucleotide polymorphism. And there are something called as variable number of tandem repeats. Now in any individual there will be certain tandem repeats. For example, GA, GA, GA. These are called as tandem repeats. Okay, it keeps on happening in one person, in any individual. These are called as variable number. One person may have say 4, I have shown here. Another person may have 6. Another person may have 8. Another person may have 2. This number of tandem repeats are going to be different in different individuals. So that is why they are called as variable number of tandem repeats, VNTRs, variable number of tandem repeats. So what will be there? The number of tandem repeats varies from one person to other, but within an individual it is unique to one individual, and the number, VNTR number, is the DNA fingerprint of that individual. The number, whether it is 2 or 4 or 8 or 6, whatever is that VNTR, that is going to be a constant for an individual, and that is his fingerprint or the molecular fingerprint, DNA fingerprint. So when cleaved by restriction enzyme, the length of the fragment will vary depending on this number. So how much big the fragment is going to be will depend upon the number of tandem repeats, VNTRs. So there are certain, such a big genome, but in that there are certain loci where this VNTR are going to be searched for. Particular probes are there which can look for VNTR in that particular gene. Loci have been identified, and those are the ones which are going to be used for DNA fingerprinting, and it can help in different disputes, paternity disputes, etc. They have been used. VNTR loci followed by RFLP followed by VNTR loci identification has been used. Now I'll go on to one more technique, and that is polymerase chain reaction, PCR. What is this? Now polymerase chain reaction, as I said, is an in vitro technique to make multiple copies of the target DNA. You want to make multiple copies for the target DNA, then we have the in vitro technique. So in this, repeated cycles of in vitro amplification occurs of specific nucleotide sequence using primers in the presence of DNA polymerase enzyme. So what is happening in this? It mimics what happens in the cells. That is, when the DNA is being copied or replicated prior to cell division, whatever is happening in the PCR also, the same things are happening. Now each cycle will amplify double the amount of DNA in the sample. So it will lead to exponential increase. Suppose this is the initial sample. In the second cycle it will form this. Each will form 2 more this. So it will lead to exponential increase, at each level, it will lead to exponential increase of the same fragment of DNA. So by the end of certain cycles only, you will get so many copies of DNA. So it is one of the easiest method. Within one afternoon, we can get multiple copies, one copy to multiple copies of the DNA. This is an exponential increase in DNA. So what are the steps in PCR? First and foremost, for this to take place, we need to construct a primer. So what is this primer? What is this primer? Primer construction, how is it done? Oligonucleotides can be sequenced and we can construct it. Now, it is essential that we should know the nucleotide sequence of short segments on each side of the target DNA. Suppose this is our target DNA and we should know certain sequences on short sequences. This is our target DNA. We need to know the sequence on this. And for this, an oligonucleotide complementary to this is synthesized. So these are known as the flanking sequences. They are used to construct two single-stranded oligonucleotides around 25 to 30 longs, complementary to the flanking sequences, and they are called as the primers. Next, what is happening? In the next section, what is happening is, first let us see, these primers have been constructed, okay? The primers have been constructed. The first thing is, this DNA has to be denatured. This DNA has to be denatured. What do you mean by denaturation? The conversion of double-stranded DNA into a single-stranded DNA is known as denaturation. So the first thing is denature the DNA. The DNA to be amplified is heated to separate the double strands into single strands. Now we add the primers. Primers have been done. We add the primers. Annealing of the primers to the single-stranded DNA. The separated strands are now cooled. When this was there, we added the DNA and they are allowed to cool. When it cools, this primer will form a complementary thing with each section of the DNA. Annealing takes place. So here what has added, before we added the primer, this was heated up, heating, then cooling, add the primer and cool it to form the complementary primers to the flanking sequences. Allow annealing to take place. Now comes chain extension. In the third step, when you have added, you allow the DNA polymerase and the DNTPs, everything are added. So when this is added, each primer is going to act as a base, act as a primer for DNA polymerase, and it will result in the formation of two strands, one having the original and the complementary, which is got from the primer, just as the way replication has taken place. So the complementary to this strand and complementary to this strand, two DNA fragments are got. So from one DNA we got two DNAs. What is done? First heating, then adding the primer, cooling, and then annealing, and then separation, adding the chain extension. So in one cycle, these are the steps that are taking place. Next, what happens? Now the DNA is again heated. The same cycle is going to continue. The DNA is again heated to again separate out. So this time there will be another DNA. Again we add more primers. All this will come and primers will go and bind. And so in each cycle there will be multiple copies of DNA being formed. Multiple copies of DNA we have got of the original DNA. Whatever we wanted, we have got multiple copies, each with its own primer. Now in this whole cycle, what is happening? The cycles of denaturing, annealing and extension are repeated. The DNA polymerase is important that this DNA polymerase which is used here should be thermostable, and so there is an important DNA polymerase known as Taq polymerase. MCQ question. So what is the use of PCR? We can amplify DNA from small amounts of starting material from variety of tissue samples. It could be a fresh tissue sample, fixed tissue sample or pathology sample. and this has revolutionized DNA analysis. The technique is simple, rapid, robust, inexpensive, and utilizes basic lab equipment, and it has been used for clinical diagnosis, molecular genetics, multidisciplinary research, etc. I'll just give you an example of clinical diagnosis. So, identification of SRY gene in ambiguous genitalia patient, application of PCR to the detection of Mycobacterium tuberculosis in sputum samples, detection of minimal residual disease of lymphoma leukemia patients by PCR technique. Now we can compare the cloned gene with the uncloned mutant, detection of low abundance nucleic acid sequences like lymphoma, HIV, etc. Forensics analysis of DNA sample. Small amounts of DNA at the crime scene are amplified. Could be from a single hair, a tiny spot of blood or semen or anything. Everything can be amplified. And prenatal diagnosis is also possible because of PCR. So for cystic fibrosis, etc., a small amount of amniotic fluid containing DNA is enough. We can amplify it. So this is usually followed by electrophoresis of the product, southern blotting, and look for migration, and we can do prenatal diagnosis. Now there are all these were techniques, different techniques. There is one more technique known as knowing the analysis of gene expression. So northern plots measure the amount and size of mRNA in a sample, particular mRNA in any given sample. There is one more thing called as microarrays. What are these? DNA microarrays are around a slide-like thing which are containing nearly 1,000 immobilized sequences. They are all organized together in one single slide. A slide will contain many different so many immobilized sequences. Okay, it will contain. Then what? In a small microscopic slide. This is usually used for genotyping or for gene expression analysis. Genotyping means we use DNA. If it is gene expression, we use complementary DNA. They are labeled with different fluorescence molecules from two different substances or two different tissues, and different colored spots are got. So depending upon the color, they tell whether gene expression is happening or it is not happening. This is known as DNA microarray. Now let us look at to summarize it. Molecular biology techniques can be used for antibody or vaccine production. How? The particular gene is isolated, put into a plasmid. The plasmid is put into a bacteria. When the bacteria divides and when the bacteria is going to translate, the protein is also going to be translated. The antibodies are produced, insulin may be produced, vaccine may be produced. All these are the type of recombinant DNA technology. Can we use it to identify disease? Yes, we can use to identify whether there is a mutation that has taken place in a particular gene or not. Again, we can, if it is gene analysis, we'll go for southern blotting. In southern blotting, two individuals can be compared and we come to know whether restriction fragment length polymorphism, is there any RFLP changes or is there any VNTR changes or single nucleotide polymorphism changes have occurred, may be done to look at the non-coding regions. In the coding regions, exactly where the mutation has taken place, we can have a specific restriction endonuclease. For example, if we want to look at HPS gene, whether the mutation has occurred in the HPS, we can look into it by looking at a specific restriction endonuclease. If that mutation is there, the fragment length is different. If the mutation is not there, the fragment length is different. So we can identify HPS prenatally, antenatally, using amniotic fluid, using chorionic villus sampling. We can also use for that is mutational analysis. We can use it to diagnose, for example, PCR can be got from a sputum sample to look for little samples of Mycobacterium tuberculosis. So even if a small amount of Mycobacterium tuberculosis is there, it can be amplified by PCR and this can be detected to know whether the person is having tuberculosis or not. So it is being used in routine labs for sputum analysis for Mycobacterium tuberculosis. We can also use it for gene therapy. I have not touched much on this. Now, gene therapy was one of the therapies that was used for severe combined immunodeficiency syndrome, SCID. That is where the experiments are going on. What they did was, the adenosine deaminase gene was deficient. They put the gene and isolated the WBCs from the individual, inserted this gene and allowed that to go back to the marrow and multiply and all that, and so that the T lymphocytes and B lymphocytes can help. But there have been cases of it was successful in, supposed to be in 9 out of 10 cases, but there have been cases of lymphomas and leukemias following the insertion of the normal gene. So it is still in experimental stages. Drug development, definitely any drug we can try to produce those drugs, and this was the best boon that has happened to diabetics, the formation of human insulin. Human insulin has decreased so many side effects which are apparent with insulin therapy, like porcine or bovine insulin. All that have decreased because of the recombinant DNA technology. It is also very simple. Just take the insulin gene, put it into your plasmid, put the plasmid into your bacteria, will divide at the same time. It will produce, translate it and produce insulin. It has been also used for anthropology analysis. Human research, genome research has been taking place, and in forensics. Everywhere, these are the different techniques that have been used for different purposes, molecular biology techniques. So with this, we come to the end of whole of molecular biology, everything about molecular biology. Thank you.